started with the planning stuff in your places, but there's going to be a lot going on, and we're very happy to have Dr. McDowell here from Virginia Tech, who's going to introduce you to some of what it is.
I think it's fine here now.
About the Arabidopsis plant. A few things for house cleaning in the meantime.
I might.
We need all phones to be turned to silent or off right now, and we need them to be away. I will free any phones I see out during the presentation today. During lunchtime, fine to do that, but right now they need to be out anyway. So thank you for doing that. We also have some other things that we will be doing today a little later, getting the handbook to you. But we really want to get into this presentation now. Oh, lost you right there. Okay, thought you'd left already. So I'm not going to give you a whole lot because this is your teacher and Dr. Dell's project. So I'm going to kind of let them go ahead and get started. Mr. B, anything you want to say before?
No, just one thing, and my students already heard this. The better job you do listening and taking notes today, the easier time you're going to have starting your actual research paper. I don't know if you've talked about the first part of that paper, your background information, but that's really what you're getting today. So take good notes. If you have questions, write them down so you can ask them later. Just pay attention. Like I said, it'll make your life a lot easier. This experiment can be kind of difficult to do if you don't understand the context of why we're actually doing it. It's like, why are we growing this plant and what are we even doing? So that's really what you want to get from today is what's the significance of what you're about to be doing for the next couple months. So like I said, pay attention, take some good notes. If someone in your group is not paying attention, you ask them to do that. So
not too hard.
Not too hard. Right, no blood. You don't want to clean the floor. All right.
All right. Without any further ado, we're going to welcome Dr. John McDowell from Virginia Tech, who's going to talk with us about the Arabidopsis. Thank you.
Good morning, everyone. Can you hear me okay in the back? Very back? All right, cool. No? Does anyone have a mic?
All right, bring it back, guys. Take two.
All right, how's this? Whoa, is that too loud? If I do it like that, am I killing ears? All right. Good morning again.
Good morning again.
So, like Mr. B said, I'm here to provide some context about the experiments that you guys are going to be doing. And really the most important aspect is shown right here in this slide. So what do you think I'm showing? What does this look like to you? Yeah, that's right. These are super sick. Any guess on what kind of plant is being shown here on the screen? It's not Arabidopsis. It's way too big for that. Soybean, good guess, but no. It's something that you guys ate maybe today, most definitely by the end of the week. Tomatoes is close. Potatoes, yes. These are good old spuds. And the disease that I'm showing here in the foreground is called late blight. I'll tell you a little bit more about it later, but first I want to tell you guys some general factoids about agriculture and why it's important. But first I want to start off by asking you some questions. Okay, so we're talking now a lot about global warming and climate change and things like that. And we're talking about how water will eventually become a really limiting resource on Earth. Water. How much do you think of the world's water is used by agriculture? Give me a guess. 1%. How much? 1%. Okay, another guess? 10. Anyone want to go higher? No one wants to go higher? 75. That's pretty much right on. 70-ish% is the estimate. Okay, what about greenhouse gases? What percent do you guys think comes from agriculture? Let's have some more guesses on that. It's not 75. Good thing, all right, 10%. 1%? There's a vote for 1%. 100%? No. About 25 to 30. Yeah. So agriculture is a big deal. And so what my group is really interested in, what gets us out of bed in the morning is how to make agriculture more productive with less input. And so the particular problem that we're interested in is loss of crops to diseases like that late blight disease that I was showing on the first slide. And so the metaphor that we like to think about for how these pathogens cause disease is that they kind of have to act like bioterrorists, because every plant has a really sophisticated immune system, just like you and I do, that can screen out a lot of pathogens and stop them cold in their tracks before they can get a foothold and cause disease. But there are some pathogens that are really good at avoiding this immune system and making their way inside the plant and then sucking out all of the water and the sugar and the minerals and all the other nutrients that they need to feed themselves and grow. And, as you can see in this slide, that kind of growth typically happens at the expense of the plant. And so, the end result for this in terms of agriculture is that it costs farmers billions on a global basis every year. And it's generally estimated that, if somehow you could eliminate all of these losses to disease, agricultural productivity would bump up on the order of 15 to 20%. That would be a pretty big deal. So, I mentioned this late blight pathogen earlier. And it's probably one that you guys have all heard about before, because it was the disease that was responsible for the famine in Ireland that killed over 1 million in the 19th century. Do any of you guys have recent ancestry that can be traced back to Ireland? I know several people who, they trace their current residence in the U.S. back to the potato famine. Their great-great-grandparents or whatever immigrated as a result of this disease. And so this is an example of how a single disease can have really significant geopolitical impacts. And even today, it's estimated that this disease alone causes on the order of $6.5 billion in damage, and that's billion with a B. And to make that real, if you could buy $6.5 billion worth of potatoes at current market value, you can meet the caloric needs of the entire world for about 2.5 days. And that's just one of dozens of diseases that cause real damage on farms. Now, also diseases that have ecological impact. An example of that is a disease called sudden oat death. This is a disease that first emerged on the west coast of California, and it's now spread to Europe. And it also threatens the East Coast because this particular pathogen species is related to the pathogen that causes the potato disease, but it has a much broader range of plants that it can affect. It can infect lots of different hardwood trees, and then also several types of shrubs that are typically used in landscaping. And so, you know, the fear is that somehow, okay, this disease will spread to, let's say, a commercial landscaping operation on the West Coast that sends plants all over the country, and then it'll spread in that way. So, you know, this is another example of a disease that that could have really big economic effects in terms of how it affects the forestry industry. So hopefully by now, I've convinced you guys that it's worth spending some time and effort to think about how to control these sorts of diseases. Now, you know, the discipline of plant pathology, you know, which is research about how to control plant diseases, has existed for about 1.5 centuries now. And the people who work in that area have come up with a lot of good solutions to mitigate the effect of disease. And these generally fall into 2 classifications. The first is chemical control, what you guys think of as fungicides or pesticides. And then the other approach that's typically taken is to use genes in the plant itself to provide resistance. And that's what we're going to be talking a lot about in a few minutes. But first, let's consider chemical control. Okay, check out the picture here. What you can see is on a tractor. He's going through his field. This looks like lettuce to me, so I'll bet that that he's spraying to control a downy mildew disease that's very closely related to the pathogen that I work on in my lab. So that's all fine and good, but what's wrong with the picture? Take a close look. Any ideas? In the back. Yeah. So that, you know, the field has to be configured in such a way that he can go through with the tractor and apply the chemical. It's probably not the most efficient use of the land that he has available. That's a good point. Anything else? Yeah, they're wearing protective clothing. At least the guy who's driving the tractor has on a, you know, protective clothing and a breathing apparatus. The guy on the back, who I presume to be the farmer's son, has a football helmet on. I guess if it falls off the back of the tractor, then maybe he'll be okay. Yeah, so, you know, broadly speaking, there are environmental effects of these chemicals that ain't so great. Any other ideas? Yeah. He is, yeah. Yeah, I mean, you know, you guys can tell just by the appearance of the apparatus that isn't very efficient either. My tires flat. Kind of looks that way, doesn't it? It may just be a pothole. Hopefully not. Okay, so you guys did a great job with that. So, you know, I think the take-home point from this is that, okay, you know, I'm not broadly disrespecting chemical controls. They have a long history of really effective use, but there are all of these problems that you guys did a really good job of describing. And another problem is that because a lot of the, you know, the most effective chemicals have bad effects for our health or bad effects for the environment, they're now being legislated out of existence. And that puts the farmer in a quandary because all of the pathogens are still there. Another big problem is that pathogens like late blight are really good at evolving mechanisms that enable them to resist the chemical. Just like, you know, antibiotic-resistant bacteria that you and I have to worry about whenever we visit the hospital. So, you know, this is not a long-term sustainable solution. And so that brings us back then to this other option of using genetics in the crop itself to provide better resistance. And that's really what, in our group, our research is all about. And so that brings us to Arabidopsis. So my group does a lot of really basic fundamental research, and we like to use Arabidopsis as a model organism for plant pathogen interactions. And there are a lot of reasons that we like to do this, but the main one is that Arabidopsis, as you guys have already seen and will see more soon, is a relatively simple organism. And so the logic behind using a model system like Arabidopsis is kind of the same as it would be, let's say if you were trying to learn how an internal combustion engine works. Imagine that you were starting from a position of complete ignorance and you wanted to learn about the basic principles of internal combustion. Well, does it make more sense to study that, learn about that in the context of a relatively simple lawnmower or a very complicated stock car with all of the ancillary stuff that you need to have on there to win a race? You go with the lawnmower. And the principle to keep in mind here is that every aspect of internal combustion itself can be applied directly to understanding how a more complicated system works. And so that's the reason that we worked on Arabidopsis. So there's a picture, a schematic of what you guys will soon see as a full-size Arabidopsis plant. And so the point that I want to emphasize first is that even when fully grown, the Arabidopsis plants aren't that big. They'll grow to a height that's more or less the same as this can of Red Bull. You guys will become intimately familiar with that later as you're counting seed pods and things like that. But for guys like me, that's a big advantage because we do a lot of genetics, and to do genetics right, you have to be able to grow hundreds or thousands of plants. And so because of the small size of Arabidopsis, we can do that easily in the relatively small space that's available in my research building. We don't have to worry about securing a field plot and all of the hassle that goes into traveling from my research lab out to the farm and worrying about the weather and all of that other stuff. We can do all of our experiments in the lab, and that's a real help. Another thing is that, and you guys will see this too, Arabidopsis grows fast. From an intact seed to a seed-producing plant takes roughly about 2 months compared to several months for most crops, let alone trees. So again, logistically, that's a big advantage for us because it enables us to do more experiments in a given amount of time. And that's very important because, okay, all of our research depends on my ability to get federal funding. And that's a highly competitive arena. I'm competing against other researchers that also have great projects. And so it's kind of a race to see who can get the most done in the least amount of time. Another really cool aspect of Arabidopsis is that it's very, very easy to modify genetically. I could take any of you in the room right now and teach you how to do this in about 20 minutes. It would work the first time you tried it, and it would work every time you tried it after that. I mean, it is literally totally simple. And in that respect, Arabidopsis is different from every other plant that's been examined. And again, in a research setting, that's a huge advantage for us. Okay, so before we move on, you guys have any questions? Okay, so I introduced you to Arabidopsis, and what I want to do now is introduce you to the pathogen of Arabidopsis that causes disease in Arabidopsis populations that exist out in the real world. Remember, we think of Arabidopsis as a model plant, but it's actually a real plant, and it has its own niches in the real world. It's typically found in Northern Europe. It actually does exist in some places here in Virginia. Its center of origin was in the Himalayan mountains. And it's found pretty much on a global basis. And so what I'm showing you here in this picture is the most commonly encountered disease of Arabidopsis. It's called downy mildew. And the reason that it's called that is for these fuzzy things that are on the outside of the leaf. Does anyone have any idea what those might be? Yeah, spore-producing structures. Each of these guys is like a little tree that produces dozens of spores. And so those spores twist off, they get blown by the wind or carried by rainwater onto new plants, and then the disease cycle can begin again. So the name of the disease is downy mildew, and the name of the organism that causes this disease is Hyalopyronospora arabidopsis. Every time I say that, I wish I had one of those little bouncing balls that jumps from syllable to syllable, because it's really a mouthful. So what I'm going to do for the rest of the talk is refer to it by its nickname, which is HPA. So we were just looking at the outside of a leaf with downy mildew, and this picture shows what's going on the inside of the leaf. And the most important aspect are these thread-like structures that you can see in the middle of the leaf. And those are structures called hyphae, and they represent the pathogen. And the way that this pathogen causes disease is that, okay, I told you guys about the spores that can spread through the wind or whatever. They land on the surface of the leaf, they attach themselves, they make these sticky structures that adhere to the surface of the leaf. And then the spore produces basically a thread that grows past the first layer of cells in the leaf into the middle of the leaf and starts snaking in between all of the cells in the so-called mesophyll layer. The important thing about the mesophyll layer is that's where photosynthesis takes place. The plant is making food, and now the pathogen is putting itself in position to take that food to fuel its own growth. And the way that we think it does this is by making these projections. Now, the buzzword that we have for these is haustoria. Now, that's a complicated sounding name, but what I want you guys to do is think about these haustoria essentially as feeding structures or even siphons. So what these haustoria do is that they make a hole in the cell wall that surrounds the plant cell, and then they push to the inside, and somehow they force the plant to make new cell membrane around the outside of the haustorium. And while all of this is going on, the pathogen is making proteins that it sends to the outside and the inside of plant cells to reprogram how the cell works. And we think that as part of that program, the pathogen is somehow reconfiguring these cells in such a way that it's easier for the pathogen to get access to the food materials that it needs to fuel its own growth. So from a cellular and a metabolic perspective, that's how we think about how this sort of interaction is working. And we think that this AP2 mutant that you guys are going to be doing experiments on is a key aspect of that process. Any questions about this? Thing to keep in mind is that, okay, these are structures that we think are enabling the pathogen to suck food out of the plant so that it can eat itself. Another aspect of this interaction is that HPA as a species is completely dependent on living plant cells as hosts. So unlike a lot of other microorganisms, it's not possible to grow HPA on a petri plate. So somehow its metabolism is intimately linked with that of its natural host, which is Arabidopsis. A few years ago, we sequenced the genome of HPA. So you guys have heard about the Human Genome Project. Well, we had a HPA genome project. And one of the things that we learned from sequencing the genome is that this particular pathogen species has lost genes that enable it to take nitrate and sulfate from the environment and use that as a food source. These are genes that are pretty highly conserved amongst a lot of biological lineages, but they're absent in this particular species. And that was interesting to us because it gave us a clue as to how the plant gets what it needs to eat from its host. And so what we think is that, okay, if the pathogen is not taking nitrogen and sulfur in an inorganic form from the environment or from the inside of the host, then it must be getting those two elements in organic form. And the most potentially accessible source of those nutrients are amino acids that the plant is making itself. And so then this is another point. The hypothesis that's guiding the work that we do on AP2 is that, okay, we think that amino acids synthesized by the plant host are a really important source of nutrition. And we also think that as part of its disease program, the pathogen is forcing the plant cells to secrete these amino acids in a place where they are more accessible to be taken up by the pathogen. And we think that AP2 is somehow playing a role in that process. Any questions about that? Nick?
So when you say that it's maybe advantageous, is that just because the pathogen just doesn't have to use resources to do it on its own anymore?
Yeah, the chemical reactions that you need to assimilate inorganic nitrogen and sulfate are really expensive from an energetic standpoint. They take lots of ATP. So if you've evolved to the point where you can take these elements in organic form from a source, then it's a lot cheaper from the standpoint of cellular economy. So from that standpoint, this, the hypothesis makes sense. That's a good question. Any others? I thought I saw a hand or two go up. You guys have gone dark. Is it just that I'm doing that good of a job of explaining things? I don't think so. Yeah. All right.
Question.
Yeah.
So what did you mean when it said on that second bullet point that was in the middle of the previous slide about transporting?
Okay, so I'll talk a little bit about that in a few minutes. So what I'm going to tell you is that plants have a very elaborate machinery for moving nutrients around inside the plant, like from the leaves to the roots, or from the roots to the leaves, or the leaves to the seeds. And we think that the AP2 gene is a key component of that machinery. And I'll explain what we think it's doing in just a few minutes. Good question. Any others? All right. Okay, so we made this hypothesis, and like scientists do, we began to gather evidence to enable us to determine whether the hypothesis is true or false. And one of the first things that we did was to look back at experiments that have surveyed the activity of every Arabidopsis gene during the course of disease of this pathogen. And this is a pretty complicated slide, and it's probably not worth either of our time for me to take you through every aspect, but the bottom line is that when you look at gene activity, what you see is that in plants that are susceptible to infection by this particular pathogen, there is a lot of reprogramming of genes that encode the machinery that underpins nitrogen transport. You don't see that in plants that are resistant to this pathogen. So what that suggests is that somehow either the plant on its own or in response to some cue by the pathogen is reprogramming all of the machinery that it uses to move this particular element around inside the plant. Now, the next thing that we did was to take mutants in which genes that encode key components of nitrogen metabolism and transport and test whether there's any difference in the interaction with HPA in those mutants compared to a wild type plant. So that sort of experiment is exactly analogous to what you guys are going to be doing in the classroom. So we took mutant plant in some of these genes, and we infected with a pathogen, and then we compared the amount of pathogen reproduction in those mutant plants to a wild type control. And the way that we did this experiment was to infect a whole bunch of plants with HPA and then count the infection structures on the outside of the leaf, just as I was showing in that picture a few minutes ago, the little spore producing things. So we do that for dozens and dozens of plants, and we take the average and we plot it on a graph, just like you guys are going to be doing with some of the aspects of the plant that you'll be measuring later on. And so then to start with, here is a control plant. And the name of this particular strain of Arabidopsis is Columbia, because it was identified in Columbia, Missouri. And so this plant, this plant, this plant, and this plant have mutations in different amino acid transporters. And so what you can hopefully see by comparing the height of the graphs is that each of these 4 mutants is less susceptible to the disease that's caused by HPA. And so then our interpretation is that somehow these genes are doing something that contributes to the ability of the pathogen to grow inside the plant. Now, a really important point to emphasize here is that these are not pathogen genes. They're genes inside the plant. But somehow these genes are doing something to support pathogen growth. Any questions about this? No, that's a really good question. These are naturally occurring genes in the plant genome, and they're what we call homologs, which are essentially copies of these same genes in every other plant for which we have a genome sequence. So these are probably components of amino acid transport that play a broadly important role in plant biology. And they're also somehow working for the benefit of the pathogen. And the one that you guys are working on, HAP2, is shown right there. So these are 4 of the genes that we're focusing a lot of our experimental attention on to understand, number 1, how they work in a normal plant, and number 2, what they're doing to benefit the pathogen and how that happens. So that's a perfect setup for the next few slides that I'm going to show. So I'm telling you all this stuff, but I really haven't explained that much about nitrogen metabolism and how that ties back with amino acid transport. So that's what I'm going to do now. Okay. So what this slide is meant to depict is that amino acids play a really central role in plant metabolism, just as they do in our own metabolism. What you guys probably all know is that amino acids are the building blocks of proteins that play important roles in the cell. And in plants, the amino acids are also important sources of carbon and nitrogen that then get switched to other compounds that are needed to make a healthy plant cell, just as is the case in our own cells. And then another important thing that amino acids are used for in plant cells is that they serve as building blocks for so-called secondary metabolites. Often these secondary metabolites do things that discourage insect feeding or slow down the growth of microorganisms like HPA. Okay, so bottom line here is that amino acids are really important for a healthy plant. And so for plants to proliferate, the metabolism and transport of these compounds has to work very well, and it's highly regulated. Okay, so this is a schematic of the plant. And so in terms of nitrogen metabolism, the way that plants get their nitrogen is typically to take it from the soil in the form of nitrate or ammonia. So those elements are taken up, and then there's a very complicated biochemical pathway through which the nitrogen and the nitrate or the ammonium is moved into amino acids. And this is something that typically takes place in the photosynthetic cells in the leaf. So plants have the ability to take up these two compounds from the soil and move them up into the upper reaches of the plant through the xylem. And that's the water conducting tissue in the plant. Okay, so the nitrogen comes from the soil, it goes to the leaves, the nitrogen goes into amino acids, and then those amino acids can be shuttled all over the plant through the phloem. Remember, the phloem is the food conducting tissue, and that is the avenue from which amino acids that are made in the leaf can go back to the root to fuel root growth, or to other leaves, or most importantly, to the seeds, which are the reproductive aspect of plant growth. And so that's kind of an overview of how the nitrogen is taken up, moved into amino acids, and how those amino acids are then moved through the plant. So did that get to the question that you're asking? And so from a genetic standpoint, then, there are dozens of genes that plants have whose job it is to control these sorts of processes. One of these is amino acid permease 2, ACT2, the one you guys are working on. So the next thing that I want to tell you guys about, to be a little bit more specific about how this sort of transport occurs, is a process that is called phloem loading. So I told you guys that amino acids are synthesized in cells that are in the middle of the leaf. And so the way that those amino acids make it from the cells in which they're synthesized to the phloem, where they're transported, is that there's specific pumps which take the amino acids from the leaf cells and pump them out, and the amino acids are then taken up